My name is Mu-ming Poo. I'm a professor at UC Berkeley. I also work at the Institute of Neuroscience in Shanghai. Today, I would like to discuss an interesting topic in neuroscience, the cellular basis of learning and memory. As you know, many of us who are attracted to neuroscience, we are attracted by the mysteries of the brain. The human brain, a 3-pound tissue that is the most complicated and most mysterious object in the universe. In this human brain that consists of 100 billion cells, each making thousands of connections with other cells, forming a complicated networks that perform all the functions that we know that human can do. Now, the brain is responsible for receiving all the sensory information that we received and integrated and interpreted it. It's also responsible for sending out the signal to execute all the behaviors, the motions, the movements of the body. The brain also can learn, can memorize, can have emotion, can be joyful or depressed. All this complex function are the most challenging problems that we are facing in understanding the brain. In fact, we can consider the understanding of brain the ultimate challenge for human understanding of nature, understanding of ourselves. So, despite this mysteriousness, there's a tremendous progress already made during the last 40 years on the cellular basis of the nervous system. The cellular molecular basis of many of the brain function is now pretty well understood. So today's topic on learning and memory is one of them. And we know what could be the site where changes in the brain could occur in association with the process of learning and memory. And I will introduce briefly what we know about the cellular basis of learning and memory, and also use some of the experimental results from my own laboratory as illustrations. So in part 1, I will talk about the Hebb's postulate and synaptic plasticity. To begin, let's take a look at what brain really looks like by cutting through the cortex, a section of the cortex, and look at the cellular organization within the cortex. If we get inside the brain, how does it look? This is a section of a rabbit cortex. If we stain the cortex, look at the cells inside, stain the nucleus of the cell, we see this massive amount of cells, highly packed cells within the cortex. Now, this is only one view of the cortex. If you do a completely different staining, which reveals the cytoplasm, the process extended from the cell, if only a few % of the cell are labeled, you will see this totally different picture. You can see individual cell body with all the process connecting each other. Now, if all the cells are staining like all the cell bodies shown here, this whole view will be black. The fact that the Golgi staining reveals only a few % of the cell allow us to see actually individual cellular morphology. Now, it is based on this Golgi staining, which was discovered by the Italian neuroanatomist Golgi in the late 19th century, that allowed the famous Spanish neuroanatomist CAS to make a very detailed map or detailed drawings of the nervous system in many different species. Now, Cahal is a very diligent, smart neuroanatomist. Here showing Cahal working in his own kitchen, on a kitchen table with a monocular microscope. He, for many years, was able to trace out, based on the fixed tissues, the Golgi stem tissues, individual cell morphologies in different parts of the brain. Here he showed his original tracing of the cortex, showing individual cells, neurons with very detailed processes. Now, Cahal's contribution was not only to draw different morphology of cells and their connectivities in different parts of the brain. He actually imagined how the brain could work. He was the one who really promoted the idea that this entire complex structure is not a continuum, it's not a continuous structure of a web like tissue, but it is made of individual cells making connections with each other. So the idea that individual cells, as shown here, is the basic unit of the nervous system, was promoted and established by Cahal. This is so-called neuron doctrine, as we know it today. Now, Cahal's important contribution is that to define the cell in the nervous system, as well as the idea that each cell has a polarity, that it has processes called dendrites that are receiving end of the cell, and a process, a very long, thin process called axon, that is the output end of this nerve cell. So the cell has a polarity. This is also Cahal's idea. Now, this polarity gives the functional definition of its morphology. And in fact, he was able to define the junctions between cells. Now we know as a synapse. That the ending of the nerve terminals that comes into the cell body or the dendrites of the other cells are forming globular structures, and he thinks these are the places where the information are transferred from one cell to the other cell. In fact, he also defined, for example, here, the terminals that just come in and making contacts with the cell body. And he defined this ending of the growing nerve terminals, as he imagined, from this fixed tissue, that these growing terminals actually making contact with its target nerve cells. So this idea that is cells communicate between through the synapse is now well established. In fact, higher resolution picture of the synapse shown here in an electron microscopy image, showing the nerve terminal, that globular structures on the cell that forming the synapse with the postsynaptic or the target cell. Now, the nerve terminal consists of many vesicles, which are packages of transmitters, chemicals, that one cell communicates with the other. Now, by releasing these chemicals at the synaptic cleft, this junction, the postsynaptic cell can respond with this signal by depolarization or hyperpolarization, a signal that can transmit further down to the cell body. So this is the synaptic structure, the key structure for cell-cell communication in the nervous system. It is also the key structure where learning and memory are likely to occur at these places. Now, how does learning and memory works? On a cellular basis, we think that it all occurs when activity are going on between cells, and it makes changes. Once the activity goes through the cell, it leaves some imprint at the junction that persists after activity is gone. So some memory trace is left in the nerve terminal or in the postsynaptic cell, so that the junction, the synaptic connection, the efficiency of this connection become changed, either strengthened or weakened. So that persistent change is the memory of the activity that has occurred. And this is basis for the cellular basis for learning and memory. So in short, activity that are associated with all the sensory, motor, and cognitive experience will leave an imprint in the neuronal or synaptic structure, change some structure we call modifications, modify the structure, and that modification will persist for a prolonged period, and that is the memory. Once that memory is stored, then we have learned, we have memory, our cognitive and behavior will be changed as a consequence. So this is a simple scheme where the neuronal synaptic changes might be the basis for higher cognitive function. So the idea sounds simple, but the person who really brings this idea together is Donald Hebb. Donald Hebb is a Canadian psychologist who have worked on the learning and memory problems, perceptual problems, using experimental psychological approaches for many years. At the end of his career, he wrote a book called The Organization of Behavior. This book has become a classic, a Bible in the field of neurobiology. And in this book, he proposed the idea how cellular synaptic function and modification can account for perceptual learning that we know in an integrated system. Now, in this book, there's a passage. This passage has been quoted again and again in all the literatures since the book was published. So let's read this passage. In one of this chapter, Hebb said that if we want to understand how perceptual memory works, how perceptual learning and memory works, we can assume this might be a cellular basis for it. Even though at that time, the basis for this, the experimental basis are not available yet. What he said is that when an axon of a cell A is near enough to excite a cell B and repeatedly and persistently taking part of firing this cell, some growth process or metabolic changes takes place in one or both cells so that the efficiency for A to excite B is increased. In other words, the synaptic connection here is affected because cell A keeps firing cell B. That's the simple passage. Now, people have used this idea for over the years, since the book was written, to come up with this very popular learning rule. The learning rule says the correlated pre- and post-synaptic activity, cell A and cell B, the activity are correlated because cell A is firing cell B. It causes synaptic strengthening or stabilization of that synapse, so the efficiency of communication is increased. And that's the learning created by the activity. And a related opposite side of this effect is that if the activity is not correlated, uncorrelated, when cell A is firing but cell B is not firing, so this uncorrelated activity will cause an opposite effect. Will cause the weakening or elimination, the synaptic context lost. And to sum these two rules together, simply, one can say that cell that fires together wire together. And this is a simple way for talking about the correlation-based learning rule proposed by Hebb. Now, to understand further, let me introduce you a little bit of the language in the nervous system. That is how the nervous system communicate with each other. The nerve cells transmit signal through a potential change called action potential, a nerve impulse. This action potential is a large depolarization of the membrane potential, so that the membrane potential go from -70 millivolts are depolarized to plus 40 millivolts. This large depolarization, transient fast depolarization that occurs within a millisecond is called action potential. This is the unit of communication in the nervous system. The neuron would fire when the depolarization reached the so-called threshold. Above the threshold, all along effects of the unit is generated, 0, 1, 0, 1 signals created in the nervous system. Now, when the action potential reaches the synapse, it transmits a signal to the next cell. This signal is mediated by synaptic potential. This is the unit of signaling at the synapse. The synaptic potential is a depolarization or hyperpolarizing potential that is caused by the activation of different transmitter receptors. The transmitter release at the synapse causes either depolarization or hyperpolarization. When it's excitatory transmission, we call it excitatory postsynaptic potential, or EPSP or IPSP. This is the unit for excitatory synapse. Most of the transmission is subthreshold in central synapse. The depolarization is below the threshold, so a single synapse cannot fire the postsynaptic cell. It's a group of synapses working together, hundreds of synapses working together to reach the threshold to fire the action potential in the postsynaptic cell. This allowed the information to be gathered from different input, and together they fire the postsynaptic cell. The postsynaptic cell serves as an integrator of presynaptic signals. Now, there's also inhibitory signal that goes the opposite depolarization. We call it inhibitory postsynaptic potential. So now today we're going to use these terms to understand how synaptic changes might be used to account for the learning and memory process within the neural network. The biggest breakthrough in this field after Hebb's proposal in 1949 occurred in early 70s. Bliss and Lømo, two scientists, discovered a very striking phenomenon called long-term potentiation. In this paper, now real classic in physiology, published in Journal of Physiology in 1973, Bliss and Lømo showed in a region of the rabbit brain called hippocampus. Hippocampus is a region in the mammalian brain that is responsible for formation of memory. If a stimulation, a high-frequency stimulation is applied to this region of the rabbit brain, the synaptic transmission will be greatly enhanced after this stimulation for a prolonged period, days and weeks after the stimulation. Now, this transient activity-induced changes, prolonged changes in the brain, is exactly what we would hope for to be the trace of memory, because memory is the trace of experience. Experience is carried by electrical activity. So the discovery of LTP, the long-term potentiation in the hippocampus, provide a clear evidence that activity can leave a long-term trace in the brain. And where is the trace? The trace is in the synapse as a potentiation, as an enhanced transmission of efficacy at the synapse. This important discovery revolutionized our thinking of how learning and memory could occur in the brain, and it directly fits along the line of Hebb's postulate that it is the cellular change at the synapse that causes the perceptual learning. Now, in LTP, now we all know a simplified term of long-term potentiation. This LTP has a very interesting property that is input specific. So look at this hippocampal neuron. which are receiving input from two different sources. You stimulate two different sides of the hippocampus. The dendrite that receiving information from input 1 or input 2 from two sides. When the high frequency stimulation is applied in one of the input, that causes a increase of synaptic transmission, shown here as the changes in the amplitude of EPSP. Now we just said that EPSP is the excitatory synaptic potential. It represents the strength of the synapse. The amplitude of each EPSP is shown as a dot here. So with time, you see this is a stable synaptic transmission for during this period. And then when a high frequency tenderness is applied, 100 hertz for 1 second is applied at this time, the amplitude jumps up. The synapse become much more efficient in transmission. You get a higher EPSP amplitude, and that lasts for a long time, shown here over 40 minutes, but in Bliss and Lømo's experiment, many, many hours, you can still see enhanced transmission. Interestingly, this has only happened in the input that you are stimulating from one side. If you stimulate from another side that activate a different set of synapse, there's no change after stimulation of input 1 has no change on the input from the other side. So the potentiation only occurred at those synapses that were stimulated. Now this is exactly what Hebb was talking about, right? Hebb's idea that cell A keeps firing cell B, and that only the connection between cell A and B are strengthened. It's a specific modification of those synapses. Other synapses associated with cell B will not be changed. Other synapses associated with output of cell A will not be changed because there's a specific modification due to the correlated firing of cell A and B. So this specificity is an important element of memory because we want the memory to be stored in specific site. So there are different sites for different memory, and the specificity allows this specific storage of the memory. There's another interesting property of long-term potentiation. It's called associativity. Now, imagine there are two different inputs coming into a hippocampal neurons. One weak input and one strong input. The weak input activates a small number of axons. Strong input activates larger number of axons. When the repetitive high-frequency stimulation is applied to weak input, it has no effect in potentiated synapse. Not enough synaptic activation happens. So there's no potentiation. If you activate strong input with a high frequency, you can produce a potentiation. The synaptic amplitude of EPSP goes up because there's a sufficient activation of postsynaptic cell to create a pre- and postsynaptic firing together. And so this is a strong input produces LTP, but a weak input produces no LTP. However, if you associate the weak input together with the strong input and fire them together at the same time, after this high-frequency stimulation, the weak input now become potentiated because by having association with a strong input, the weak input are helped to produce its potentiation because the postsynaptic cell could fire at the same time while the presynaptic cell is firing. Now, by itself, it cannot fire the postsynaptic cell, but a strong input helps to fire the postsynaptic cell. Right? So the associative learning is a very important property. It's important because we know associativity is an important element of many learning process. In a classic example of Pavlov, the classical conditioning, where the ringing of bell is paired with the presentation of a meat, the dog salivates as a result of ringing the bell alone. The bell alone by itself usually is a weak stimulus, but after the pairing with a strong meat signal, the bell would be able to trigger the salivation of the dog. Right? So this associative element of the Pavlovian conditioning has a cellular basis at a synapse. Finally, in the late 1890, early 90, in the hippocampus, one also discovered the phenomenon of long-term depression. This is an opposite effect from the LTP, we call it LTD, the long-term depression. In this case, LTD was generated by a low-frequency stimulation of the input. When repetitive low-frequency stimulation, after a while, the synaptic strength are reduced and persistently reduced for a long time. That's called LTD. And these effects, again, synapse-specific, a different input that was not stimulated will remain unchanged. Now, LTD was originally discovered by Masao Ito at Japan in the cerebellum. There, one show a repetitive stimulation of the input to Purkinje cell from parallel fiber, also show a persistent depression. So in both hippocampus and the cerebellum, there are changes at the synapse that represent a persistent reduction after activity. So now we have a very useful mechanism that activity not only can potentiate the synapse, it can also reduce the synapse. It's a bidirectional change of the synaptic strength. But through these changes, one can store information associated with the activity. So what is the basis, the molecular basis of long-term potentiation and long-term depression? The presynaptic activation releases glutamate at the excitatory synapse. Glutamate activate glutamate receptors, different type of glutamate receptors. Some of them allows the calcium to flow in. The most important glutamate receptor involved in this long-term change in the synapse is called the NMDA receptor. NMDA receptor is a special type of receptor which are normally blocked by magnesium, but when the postsynaptic cells are excited, membrane are depolarized, the magnesium are kicked out of the channel. The calcium can flow into the glutamate receptors, create a large amount of calcium. Now, the calcium that flow into the cell depends on the pattern of activation of the postsynaptic cell, pattern of release of transmitters. During the induction of LTP, there's a large depolarization of postsynaptic cell, and there's a transient high-level calcium flow through the NMDA receptors. You have a large amount of calcium transient that will be able to activate a group of kinases that are calcium dependent. These kinases would act at a postsynaptic cell to create LTP. On the other hand, a different pattern, a low frequency stimulation, a low level of depolarization of the cell, create a lesser amount of calcium flow that can only activate phosphatase, an opposite enzyme from kinases. It dephosphorylates proteins, and that phosphatase action leads to LTD. So the same calcium signal with a different pattern create different consequences in the cell, cause the potentiation or depression of the synapse. Now, how does this work? In terms of changing the synaptic strength, it is generally agreed now that there are most likely changes in the postsynaptic MPA receptors. The amount of receptors in the postsynaptic membrane can be increased or decreased following activity that leads to LTP or activity that leads to LTD. And there are also evidence that the release of transmitter, the amount of transmitter release can be potentiated, can be increased So even though the receptor may not be increased, if the transmitter release is increased, you can also potentiate the synapse. And reduction of transmitter releases can also depress synapse. Thus, depend on different type of synapse and a different experimental condition and different developmental stages, one might have a different forms of LTP and LTD that depends on the postsynaptic modification of receptors or presynaptic modification of transmitter release. And in the consequence of all this is the change of the efficacy of transmission through the synapse. So now, we're knowing that individual synapse can be changed by activity. Where are these synapses in the brain? Where is the memory trace in the brain? The person who brings this question sharply in focus is the famous American physiologist Carl Lashley. Carl Lashley was concerned about where the memory is stored in the brain. He has performed a long series of studies to investigate this problem. And one straightforward approach, he thought, is that if one destroys different parts of the cortex, if the memory of a rat, after specific destruction of the cortical areas, if there are certain damage causes the rat to lose its ability in running the maze, and the memory that's stored during training of the maze training will be in that area. So by systematically delete or destroy different parts of the rat brain and test the rat's ability of run maze or learning the maze and memorizing the maze experience, he found that he was hoping to find a specific area where the memory for the maze learning is stored. Well, in the end, his long series of experiments was not very, it was quite disappointing. What he found is the following relationship, that the number of errors that the rat would make is directly proportional to the amount of the tissue, cortical tissue, that was destroyed. In other words, it appears everywhere in the cortex contribute to the maze learning. And this disappointing conclusion led Lashley to conclude that memory is equally distributed in all cortical areas. Now, this idea only is partially true, because at the time where the experiment was done, so that the cortical areas that are not necessarily directly contribute to the learning or memory, but contribute to the process of running a maze are also affected. So it's hard to make conclusion that all the cortical area are actually involved in the learning, in storing the memory. So the current view actually is partially in agreement with Carl Lashley. That is, the memory is distributed in many areas, but it's not equally in all areas. There are specific areas to store memory, specific memory. Now, exactly where they are, in fact, is still not quite clear. This is an ongoing problem in neuroscience, exactly where different memory are stored. So the conclusion is that even though we know that the changes can be created at a synapse that can be a unit or the basis for memory storage, but exactly where that synapse is, or what groups of synapses are involved in storing the memory, remains unknown for most of the memory we know. So finally, I want to go back to the Hebb's postulate. Now, Hebb's idea is not only that the cells are storing memories at a synapse, So what synapse he's talking about is the synapse he's talking about are synapse made by a specific group of cells that are activated by the sensory experience. In fact, his synaptic hypothesis is part of a larger hypothesis we can call cell assembly hypothesis. Now, in this hypothesis, he said that the synapses are strengthened by cortical activity, which we know, the learning rule. The cell fires together, wire together, created by the sensory input. But the perceptual memory of sensory experience involved the formation of a specific group of interconnected cells he calls cell assembly. It's the synapse made by this cell assembly that are strengthened through firing together. And this is the idea of Hebb's, that the origin of the Hebb's postulate of synaptic learning. Now, we can illustrate here with a simple drawing. This was a nice drawing made in Mark Bell's textbook. Here we have a group of cells interconnected by synaptic connections. Let's represent the external stimulus with a ring. This would activate specifically this group of cells. When they are activated together, they fire together, their activity causes the strengthening of their connection, so they are highly connected. Strengthening of connection between these all cells, this specific cell assembly. Now, in Hebb's postulate, he also said that after the experience, there's actually a reverberating activity, that the activity still keeps going on within this group so that this firing can persist to strengthen the connections. And that reverberating activity is, again, an idea that Hebb's trying to promote. And this is an idea that will come back in a later part of this lecture. So after this assembly, this connection has been strengthened among this assembly, a partial external cue, not exactly a ring, but only a part, could activate a part of the cell, a part of the assembly, but because of their strengthening of connection among all these cells, all these cells within assembly will be activated as a result of strengthened connections. So partial cue now can record the original image. And there's a memory stored in the assembly that can be recalled by a partial stimulus. Right? This is the basis for Hebb's cell assembly, the idea of perceptual learning through synaptic connection. Okay, so this is the end of the first part, but before the end, I want to show you an example of perceptual learning. So for those of you who have never seen this picture before, can you see anything out of this background of black dots? You may not. But if I point out to you that there's a dog, a Dalmatian dog, right here, with the head and the body. Now, once I point this out, you look at it for a while, you'll remember there's a dog. So next time when you look at this picture, you immediately can see this dog because the perceptual experience have left a memory in your brain. Any partial stimulus here will be triggering the complete image of a dog that in your brain. Right, so this is an analogous situation when we talk about cell assembly, how cell assembly idea can be used to explain perceptual learning.